Drug Development Overview, prepared for and presented at the 2014 Square One Entrepreneurship Training Program. Square One is a program of the Center for Emerging Technologies. CET is an affiliate of the Cortex Innovation Community. Square One is funded in part by the Missouri Technology Corporation.
If you understand this process, you'll catch on to the other regulatory programs for the other drugs or the other products that are regulated by the Center for Drug Evaluation and Research. Okay. In a lot of cases, I'm going to take a business strategic point of view on this. And here's a view from 30,000 feet. Development timeline for new drug development and approval is 10 to 15 years. From the inception, from the time that the sponsors involved in discovery to the point of submitting the NDA or BLA at the end, it's got a 1 in 5,000 chance of success. And if he carries it that far, the development costs are going to be $1 billion plus on average. And even though there have been a myriad new technologies now available, very sophisticated, very advanced, they've done tremendous things in the lab and helping to help us much better understand disease. But despite all these new technologies, they've added significant cost to the development process, but have yet to improve the productivity. Productivity measured by the number of new products being approved every year. It's still running about the same, despite all the new technologies available. So, I think by any measure, this is a high-risk poker game. Now, there's a drug development process from drug discovery through to FDA review, final approval, and commercial manufacture. And you can see the odds starting at 5,000 in discovery phase, winnowing down through preclinical to 5. And of the 5 drugs that will go into clinical development, human testing, 1 will on average be approved. So you got a 20% chance from the time you enter the clinic. Before we get into the process, though, let's take a quick look at FDA, who designs the process and regulates the process for drug development. Just a quick history. Just to give a little context, FDA history, 1906, Pure Food and Drug Act was passed, a lot of contaminated foods, and also contaminated misbranded drugs, cocaine, opioids were routinely used without requiring labeling. So first legislation was in 1906, Pure Food and Drug Act, which prohibited interstate commerce of misbranded and adulterated food and drugs. It was a minor step, and this was being regulated by the USDA, Department of Agriculture. 1930, the FDA actually got its name, the Food and Drug Administration, still under the USDA, but still with very little regulatory authority. That changed in the late 30s. And in particular, what changed it was a public furor which arose over the marketing of a toxic sulfanilamide elixir. Sulfanilamide was a sulfa drug. Sulfa drugs were the early antibiotics and anti-infectives before the invention of penicillin and the modern antibiotics, and they're derived from coal tar. And this sulfanilamide product was a pediatric formulation, liquid formulation. It used a highly toxic solvent for children. Over 100 children died. There was a public uproar, congressional hearings, And the outgrowth of this was that, amongst other important tenets, that for the first time, drug manufacturers had to demonstrate safety prior to marketing approval. So for the first time, FDA had the final authority on providing marketing approval, on safety. They had to show safety. So this is the first requirement for any kind of clinical studies to demonstrate safety. It also authorized factory inspections. This legislation, this is still the core legislation under which FDA operates today. It's commonly called the FD&C Act or just the Act. One other important, one important amendment, one of many, which occurred in 1962, result of another public outcry, thalidomide. Has anybody heard of thalidomide? Well, back in the 1960s, it was being developed by a small German company as a sedative. It was a highly effective, very mild sedative, but it also is being used a great deal to treat the nausea of morning sickness. Unbeknownst to the developers at the time, the drug was passing the blood-placental barrier, and lo and behold, within 2 years, some horrendous birth defects began to emerge. Public outcry, development was stopped in the United States, and the result was the Harris-Kefauver amendments, the FD&C Act, which required for the first time that now manufacturers had to demonstrate efficacy as well as safety. So the modern era of clinical development of studying both safety and efficacy emerged and had to be demonstrated to FDA prior to gaining market approval. Okay, FDA today. The head is the Office of the Commissioner, Margaret Henberger. She's been there since 2009. She has many offices and centers reporting into her. The one we're interested in is the Office of Medical Products and Tobacco. Sort of interesting bedfellows there. But under the Office of Medical Products and Tobacco are several of these centers. The center we're interested in is this CDER, Center for Drug Evaluation and Research. That is where all drugs, new drugs and NDAs are reviewed.
And just to clarify, that's synthetic chemicals used as drugs, small molecule drugs, right?
No, it's also biologics.
Oh, okay.
Yeah. That was changed in 2002. Biologics were over here.
Okay,
But in 2002, and vaccines are still over here.
I see.
But in 2002, they changed it over, so therapeutic biologics are here.
I'm obviously dating myself, thank you, Carl.
Pre-02. So this is it today. Janet Woodcock is the director of CDER, and here's the scope of products that are regulated. As part of FDA, CDER regulates over-the-counter and prescription drugs, including biological therapeutics and generic drugs. This work covers more than just medicines. For example, fluoride toothpaste, antiperspirants, dandruff shampoos, and sunscreens are all considered drugs under this provision. All right, so back to the schematic of the process. Now let's work our way through the process, starting with drug discovery, through preclinical, through clinical trials, FDA review, and hopefully final approval. A trip which can take 10 to 15 years and $1 billion plus. Okay, I'm going to skip that one. I'm just going to touch briefly on drug discovery. Anybody here who's in the area of drug discovery, you're probably in the wrong room. Because you should be either in a lab in academia, or you should be in a big pharma or a big biotech working in a discovery group. This discovery is really for the big boys, be it the academics or the big companies. But let's talk about it briefly. There are 4 stages to modern drug discovery. The first, actually, before they even get into the formal process, is understanding the disease. And the research group that's working on discovery has to have a very thorough understanding of the disease as far as the underlying pathophysiology and the biochemical pathways that impact upon the disease process. With that in mind, they start with target identification, looking for a target within the biological pathways that impacts the disease process and which can be intervened through drug administration, and that has a receptor that a drug can bind to. This is usually some kind of a protein, a gene, a hormone, an enzyme. And it takes several years and a lot of research to come up with a small collection of suspected targets. Novel targets are usually being looked at to address the therapeutic against. Once targets are identified, they have to go through a validation process to confirm, in fact, that the target plays the role in the disease that is anticipated. And there's a lot of animal work, there's a lot of in vitro and in vivo work, which goes into making that validation determination. With a target validated, now the researchers shift attention to finding a drug for that target, looking for a hit. And they start by looking for a druggable lead compound, a compound that will bind to that receptor and will bind securely and selectively. The firmer and the more selectively it binds, the less chance you have of adverse reactions. And there are many techniques. One of the most common ones used here is high throughput screening, where you can get through automation and robotics, you can examine thousands of test compounds against the target to look for hits. And it's only the hits you're interested in. That means that they've bound to the target. And then the biochemists begin working on the hits to try to tailor them into an effective drug. Okay. Once the potential drug candidates have been identified, then go to lead optimization. This is developing analogs of the drug to look for even better binding. So you're trying to optimize that connection between the target and the drug to improve selectivity. And lastly, that last line says down there, file patents. You want to make sure, first of all, that no one else has filed a patent for this compound, but you also want to get your patent work started. Okay, so now the drug discovery portion in itself can take up to 4 years, $400 to $500 million. So it's significant. Yeah.
I was just wondering, has nanotechnology helped any of those steps? You know, the advancement of the nanotechnology that allows you more targeting ability?
It has in some cases, as far as being able to better deliver the drug to the site You know, there are a lot of these active biologics that are coming out that actually have to be in contact with the disease type. Antineoplastics, for example, in cancer, a lot of them have to actually make contact with the tumor cell in order to be effective. And a lot of them resort to some kind of a nanoparticulate formulation.
To connect it.
Yeah,
Thank you.
yeah. So yeah, it is an important part of the formulation. All right, so we moved to preclinical. So now we're down to about 250 candidates that may make it. And now we're doing the initial animal testing to see how the drug profiles in animals. In vitro, in vivo experiments, typically in 2 species for the animal toxicity, 2 species. And then if in fact there is an animal model for the disease, do efficacy studies in the animal model, even though in many cases, as we know, animal models are not necessarily predictive of human outcomes. Also, conduct the pharmacokinetic studies in animals, the ADME, absorption, distribution, metabolism, excretion studies. This is all in preparation for filing the investigational new drug application. You need to scale up the manufacturing to pilot plant scale, because you're going to need to be at least at pilot plant scale in order to provide clinical supplies for the next stage. And the pilot plant is going to have to be set up and operated according to GMP, good manufacturing practices, as set forth by FDA. If you're not one of the big guys, you're going to want a contract research organization. If you're an early stage, if you're moving into this area early on, you're not going to have the infrastructure, the resources. There are a lot of excellent CROs out there that can handle either certain portions of the process or everything from soup to nuts. And you can have several of them bid on it, and you can negotiate with them, and you can have them negotiate on just certain parts of the process. You don't necessarily have to pass everything over to one, although a certain amount of continuity is very useful. And most contract research organizations have some regulatory expertise as well to help in working with the agency, in addition to helping you design your preclinical and your clinical plan. All right. Well, with the right infrastructure people on board, you now prepare your investigational new drug with all the data that you've collected preclinically. And from a time and cost standpoint, we're looking at $5 million and another $1.5 million.
Carl, it's important, I think, for the class to know that the IND, that's typically you meet with the FDA before the IND submission, but really the IND is the investigational new drug application is typically the first application you make, isn't that correct?
Yes. Right. Yeah, and as Harry said, the first thing you do is you have a pre-IND meeting, which is really critical. In order to, it's a great opportunity to sit and explain to the agency what your plan is, what you're trying to accomplish, what your drug is, and to get some feedback and some suggestions on how to best lay out that plan. Because this is, in fact, the first exposure that the agency will have to your drug and to your program. Following that meeting, you'll submit the IND, which is really a request to allow human exposure to the experimental drug. And the agency, over a 30-day period, will be reviewing all your preclinical data, all the pharmacokinetics, your toxicity studies, the two species. They're going to want to know the description of the mechanism of action, adverse reactions. All this testing, you'll have to show that it's been done under GLP, good laboratory practices. You're going to need from the manufacturing side, this is typically called the CMC section, Chemical Manufacturing Controls. You're going to need to demonstrate, first show the chemical structure of the drug, the formulation, the recipe for the formulation, and you're going to have to show your manufacturing control data and assay methodology. And that's all going to have to be done in accordance with GMP, good manufacturing practices, as set forth by FDA. You have to lay out your clinical plan, good clinical practices. It has to be in accordance with. And at this case, if you really believe that you qualify, you could apply for priority review, although most drugs apply for that a little bit farther down the road when they have more clinical data to support that kind of a request. There's a 30-day review time. And if you don't hear back from the agency, you're clear to go ahead. If they don't respond, you can proceed. And the IND becomes the ongoing open file at FDA tracking the drug through the NDA process. All right, so you're for the first time.
What's your question? I'd like to ask, how much that will cost for filing, for FDA filing?
For the NDA,
Yeah.
for the IND, nothing.
Nothing.
For the NDA, 2.2 million. Well,
Yeah, but there's ways around that, we'll talk about that. Okay.
yeah, that's for a new innovative drug application, and I'll touch on that in a minute. Okay, so phase one, you can move right into phase one, 30 days after submission of the IND, as long as you don't hear that you're being put on clinical hold by FDA. Now, one other thing you always will be required to do in doing clinical trials is you'll have to get approvals from the local IRBs, the Investigational Review Boards, at each center where you're going to be conducting trials. Typically in phase one, you're going to be treating 20 to 100 healthy volunteers, because you're only doing safety studies in phase one. You're testing safety, tolerability, and it's only in certain life-threatening diseases where you're required to treat patients instead of normal volunteers, cancer being one. What you're trying to determine is safety. You're not trying to determine the pharmacoeconomics again, the ADME in humans this time, comparing it to your animal data. And you're doing some dose ranging to try to assess the maximum tolerable dose while also assessing side effects. So you're incrementally increasing the dose to see where's the point where you begin to experience some unacceptable side effects, which you can then identify as your maximum tolerable dose. Length of this phase one period is typically 1 year, cost of $1 to $10 million. Phase two, there's no need to consult with the agency prior to phase two. You are filing as you're going on, you're filing monitoring reports throughout the program, so the agency is being kept informed from the monitoring reports that are being filed. Yeah.
What if your drug is dealing with animals that's being eaten by humans?
I'm sorry, if it's dealing with animals?
Dealing with animals that's consumed by humans. What?
Food producing animals? You mean like
Yeah,
beef,
production animals, right?
beef cattle?
Yes.
So you mean if you're treating food producing animals?
Yes, if you're using a drug to.
Okay. There's a separate office for treating for all your animal,
Center for Veterinary Medicine.
yeah.
And they're, basically you have to show that the material is safe, not only for the animal itself, but most importantly for human consumption. So they'll make you measure residuals, it's called, or any residuals of the drug remaining in the meat or in milk or eggs or whatever animal products you're using. You have to show that if there is any kind of residual, it's safe. Obviously, it's best if you don't have a residual, and frequently, you'll try to design your studies with a washout period, so for a period of time prior to harvesting the animals, they're not dosed with material typically. So it generally kind of mirrors what goes on the human side. The costs, generally speaking, I'm going to make a gross generalization here, are an order of magnitude lower, but the time is actually not that much faster. It's a little bit faster, but not that much.
So you said go through the Center for Veterinary Medicine?
Yeah, it's the,
Yeah.
And then, I guess on the previous slide, we talked about institutional review boards.
So it's back under here. There's under here and out here is a Center for Veterinary Medicine.
And, again, the same kind of, you want to have a pre-meeting with them. So, and then the other thing that was mentioned is IRBs or institutional review boards, so the place where you would do the clinical trials, right? So if you're doing clinical trials with a university, so there's an animal use committee, which performs that same function within the university. Then there are contract research organizations that will do animal trials for you. And they have their own internal, veterinary group that passes on whether the animals are being treated fairly or not. They're generally more forgiving than the university setting. And then there is the possibility of doing studies with large producers. Of course, there you have to typically give up rights in advance. Okay, so the very quick summary, we could deal with some of your specifics offline.
Okay.
But most of this process is
It's similar.
very similar, very similar. Okay, so we're moving to phase 2. This is the first time that you have the opportunity to see if your drug works in man. Proof of concept, it's called. So after 4, 5, 6 years, many millions of dollars, for the first time, you get to see if this drug actually works, the one that you've been sweating on and nurturing through the development process.
Carl, we're going to have to move it along.
Okay, all right. Controlled trials are conducted in 100 to 500 patients in this phase because they're shorter term. The control group used to always be placebo controlled. More and more now in certain diseases, the agency is asking you to use the current standard of care as the control group. So you're measuring against the current standard of care. Safety and side effects are monitored, and you're doing dose ranging once again. Remember, in stage 1, you were looking for maximum tolerated dose. Now in this phase, you're looking for minimum effective dose, because now you're measuring efficacy. So then after this, you'll have a range, maximum tolerable, minimum effective, and somewhere in there are going to be the dosage choices that you make for phase 3. Results must be statistically significant versus control group. Length of time, 2 years. Cost, and now escalating from 10 to 100 million. Phase 3. Now another meeting is required. Your end of phase 2 meeting with FDA, where you review your findings from phase 1 and 2, and you set forth your phase 3 plan. The primary objective is to confirm efficacy and safety in large scales, multi-center, long-term trials versus controls. So now we're talking 1,000 to 5,000 patients. And you're having to select your final dose or final dosage. You're measuring long-term efficacy and safety over a period of 4 to 8 years, and you're certainly tracking side effects. All of this will impact upon your final labeling. Again, your results must be statistically significant to a high level of confidence. Manufacturing scale-up now during this phase must begin to be made up to commercial scale. And in some cases, you may even consider doing pharmacoeconomic outcome studies if you're going to, and more, this is important for manufacturers to be eligible for reimbursement. And CMS, Center for Medicare and Medicaid Services, is making the determination for all federal purchases as to whether reimbursement will be allowed. So sometimes it's worth looking into doing pharmacoeconomic outcome studies where you're measuring the impact of your drug's results and the outcome on from an economic and from a social standpoint as to how much money is it saving the healthcare system, how much is it saving society. Those can be compelling studies when trying to justify your pricing. All right, just a quick peek here. This is from FDA files, and this is just to point out that the averages for clinical trial times can be deceiving. The top bar is CNS up there, central nervous system diseases, and that therapeutic class is particularly difficult to study, and typically you're spending 8 years in the clinic. And now most of those are neurodegenerative diseases, Parkinson's, ALS, Alzheimer's. Difficult to study, long-term, costly. Down at the bottom, you can talk about anti-infectives, antivirals. It's an acute condition, it's short-term, there are very clear clinical endpoints. You know when you've eradicated an infection, you know when you have significantly reduced the viral load, so it's easier to study, easier to measure, so thereby shorter term, and you get faster approval times because the agency understands those endpoints. So it runs from 8 years for CNS down to a little over 4 years for antivirals. Clinic time. All right. Well, I'll skip the orphan drugs. We'll just do the Prescription Drug User Fee Act. PDUFA, it's called PDUFA. First passed in 1992. The problem was that FDA was taking longer and longer to process and approve FDAs, up to 3 years, and manufacturers were complaining. Our patents, the patent clock is running, and the drug is sitting there, and it could be out. We could have it out there generating revenues, but it sits. Finally, came to a compromise with Congress and came up with this Drug User Fee Act, PDUFA. It's renewed every 5 years. We're now on the fifth one. The fifth one was just started in 2012. The idea is when each sponsor submits an NDA, they pay a user fee. That user fee is specifically earmarked for infrastructure and resources to accelerate the review process. And it includes new scientists, new staff, computers, and it's been effective. The user fee in 2013 was $2.2 million. The fee is specifically earmarked to accelerate the review process, and it has been effective in providing FDA with greater accountability and with greater collaboration with industry.
Hey, Carl, I noticed you've got about a half a dozen slides left to go. I hate to rush you, but you got about 3 minutes.
Okay, all right. Well, here's the NDA, or BLA. The BLA is a biologic. It's a drug manufactured by biological manufacturing, like recombinant manufacturing. It's submitted as a biological license agreement, NDA, new drug application. That's the small molecules. You submit, and FDA has 60 days to review prior to accepting. Then they give you a PDUFA date, they call it, which is 10 months for a standard review, 6 months for a priority review. So from the time you submit, you get approval in 10 months, or, excuse me, you get approval in 12 months for standard and 8 months for priority. And in 2013, FDA met their dates 100% of the time. So they really are becoming responsive because they have to meet regularly with Congress to present on this. And you present all your clinical, preclinical data, proposed labeling, chemical manufacturing controls. You have to demonstrate that you can maintain identity, purity, and strength. Manufacturing scale-up. You have to scale up. You have a pre-approval inspection and have 3 production batches that meet specification. And post-marketing surveillance. If you get approval, you're often going to be asked to conduct some form of post-market surveillance to track some potential issues of safety or toxicity for a longer term period than the clinical trials. Okay, we'll skip that. This simply shows that the approval rates, I said it's 20%, but that in general, 1 out of 5 drugs survives the clinical trial process to approval. And you can just see that this varies by therapeutic area. Again, systemic anti-infectives get almost a 24% batting average, whereas central nervous system products about 8%. So depending on the therapeutic category, your odds are going to change as far as your clinical success. Okay. Yeah.
So, I have one question. So what I understand, the new drug development is very challenging and billion-dollar project. So what is your comments about development of biosimilar that is $1 billion in the market, and many biosimilar patent will be expired next few years?
Well, the FDA guidelines are now out on biosimilars, and there are biosimilars in development right now. I think last I read, Enbrel may be the first. Of course, it's going to be more difficult than the typical generic path because it's going to require clinical trials in addition to just demonstrating bioequivalence in normal subjects. So it's going to be more difficult, it's going to be more time-consuming, and more costly than generics. That being said, it's going to be significantly less expensive than the expense of the original manufacturer. So they're going to be a reality. In all likelihood, there will be maybe 2 competitors that'll come out for each of the original drugs. So it'll carve up the market, the prices will drop a little bit. So it's going to be an opportunity, and most of the major pharmaceutical companies, one way or another, are pursuing biosimilar
We have a
opportunities.
real expert too, by the way, a guy who runs a company called Raybridge here, he's an expert. So if you're interested in that area, I can provide an introduction. Any other questions for Carl? All right, Carl, thank you very
Okay.
much.